Hello students, so today we'll be starting with the next topic. We all know that DNA is our genetic material. The genetic material is the one that determines our characteristics. It was passed on to us by our parents. The experiments that proved that DNA is our genetic material are the Griffiths experiment and Avery, MacLeod and McCarthy experiments. Now, Griffiths experiment included two things: the Streptococcus pneumoniae and the mice. Now, the Streptococcus pneumoniae are of two types: the smooth Streptococcus pneumoniae and the rough Streptococcus pneumoniae. The smooth type is a virulent one, which means it is a pathogenic type, and the rough is a non-virulent one, that is, it is non-pathogenic. The smooth and the rough types are further subdivided into various strains like S1, S2, S3 and R1, R2, R3. The difference between smooth and rough Streptococcus pneumoniae are: the smooth Streptococcus pneumoniae contains a capsule around its cell wall, whereas the rough Streptococcus pneumoniae has no capsule around its cell. Now, in the Griffiths experiment, he first injects the mice with smooth strain, which is virulent, that is pathogenic, and the result was expected. The mice suffers from pneumonia and dies. Now the second time he again injects the mice but with the rough strain, that is non-virulent strain or non-pathogenic strain, and again the result was very much expected. The mouse lives. Why? Because the rough strain is non-pathogenic. The third time, Griffiths kills the virulent smooth strain with heat and injects it into the mice. And the result is the mice lives. Why? Because the bacteria has been killed. So it is inactivated. Now finally, what Griffiths does is he combines the rough strain and heat killed smooth strain and injects it into the mice. The result is the mice dies. Now it is very unexpected because the rough strain is a non-pathogenic strain and the smooth strain has been killed. So how does the mice dies? Griffith, after performing the experiment, concluded that this happened because the non-virulent R strain bacteria have somehow been transformed or changed into the virulent S strain. This may be due to the transfer of genetic material, which enabled the R strain to synthesize capsule and transformed itself to the virulent S strain. Now, the genetic material responsible for the transformation could be a protein, DNA, or RNA. Now, to find out this, three scientists, that is Avery, MacLeod, and McCarthy, reconducted the experiment. They separated out the biochemicals from the transformed bacteria and analyzed them. They discovered that DNA above caused the R strain to become transformed. So they concluded that DNA is the genetic material.